the amazing Steve Horvath, whose name all of you know, and who has recently shifted from UCLA to Altus Labs, and I'd be delighted to hear what he has to say.
Yeah, thank you, Aubrey. This is another phenomenal conference organized by Aubrey. But I'm here because somebody said that Irish rain is good for your hair. So, so I chose the same title, love it, Elusive Concept of Biologic Age. I will start with some questions. I don't pretend I have an answer, it's really a question. So is it scientifically meaningful to use biomarkers of aging for children? Or another way to say it is, do children undergo aging processes? And if there's aging in children, is aging actually development? You know, so I think the answer to this question really depends on the kind of biomarkers you use. What I can tell you is that in terms of epigenetic clocks and methylation changes, we do have an answer. So yes, epigenetic clocks very much apply to development. Here you see on the y-axis methylation, on the x-axis you see chronological age. Importantly, it starts at age 0. You see a curve. This curve is called a log-linear relationship. Initially, it's a log, and then later it becomes linear. And then there's quite some literature on linking the early changes during development to actually development, for example, pubertal speed, and so on. Anyway, this is all based on the human pan-tissue epigenetic clock. And just to foreshadow the presentation, we actually use the very same log-linear transformation in our universal pan-mammalian clock. So it's kind of interesting. Here are my axioms for an indicator of biologic age. But very importantly, these axioms do not talk about children at all. Why? Because I don't want to impose that axiom on other biomarker. But anyway, I start out with the obvious. A biomarker of aging must be quantitative and correlate highly with chronologic age. Otherwise, it isn't a biomarker of aging. You know. Then the other aspect is it should be connected and predictive of mortality, but it will be distinct from it. It's not simply a mortality risk predictor. The third axiom is all about whether you are dealing with a molecular biomarker. To me, a molecular biomarker of aging must relate to a fundamental characteristic of most cells, because most cells undergo aging. And of course, methylation focuses on the DNA molecule. But you know, it could be other fundamental characteristics, for example, mitochondrial function or lysosomes or something else, you know. And then the fourth axiom that is really the focus of this talk today. To me, a biomarker of aging should apply to all mammalian species because, in my view, all mammals experience aging. And I want to highlight that this is not a fact, it's an axiom, you know, so I impose it. And so you could debate it, you know. Before I continue, I want to say that we just released a very large mammalian data set, over 15,000 tissue samples, 70 tissue types, 348 mammalian species. It's on GitHub, it's on Gene Expression Omnibus. There's software available. We practice open science, so everything is open. And this data set was created by 200 individuals. You see a few names here. So Vadim Gladyshev contributed a lot, Vera Gorbunova, Andrei Selyuanov, Juan Carlos Belmonte, Emma Teeling, who is in the audience. So many, many people. And we ended up profiling 25 out of 26 taxonomic orders. Some of you may know mammals can be grouped into 26 taxa. And the one we missed has a name that cannot be pronounced, but it's a marsupial mole. So if you live somewhere in the middle of Australia, if you could send us some roadkill, we would appreciate it. You could change mammalian aging signs if you just scrape it off the road. Now, in order to create this data set, we used a particular technology, which is known as the mammalian methylation array. Its strength is that it hones in on ultra-conserved regions of the DNA. So often these regions play a role in development. And this is therefore a very nice data set for comparative biologic questions. It's actually very easy to compare one mammal to another species if you use these particular data. The drawback of the data is that we're missing the non-conserved places, right? There will be millions of cytosines that are not covered by the array that may be unique to an animal. I will briefly talk about these third-generation clocks. These are clocks that apply to multiple species at the same time. One mathematical formula, one regression model measures aging in all mammalian species. And to arrive at that, we are using the concept of relative age. Vadim Gladyshev talked about it briefly. So relative age is defined as the calendar age of the animal divided by the maximum lifespan of the species. So it's a number between 0 and 1. And so, for example, a 2-year-old rat has a relative age of 0.5 because the maximum lifespan of rats is about 4 years, so 2 divided by 4. In humans, we know the maximum lifespan is about 122. And so then the relative age of a 61-year-old person is 61 divided by 122, so it's 0.5. So this is a very crude way of aligning ages across different species. It's crude, no doubt one can do better. But what I will say, it does work, in the bottom, I show you an epigenetic clock that applies both to rats and to humans. The green dots are human tissue samples. The orange dots are rat samples. And you see this alignment. The x-axis, again, shows relative age of these species. And we, of course, started the consortium because we wanted to know, is it possible to construct epigenetic clocks applicable to every mammalian species? And I really want to highlight one more time, this is now one regression model, one formula, one set of cytosines. Roughly about 600 ultra-conserved cytosines make it into these epigenetic clocks. And here I show you Arkelou's universal clock applied to 185 species. And the right panel shows you, on the x-axis again, relative age. So although we have vastly differing species separated by 200 million years of evolution, different ecology, bats versus bowhead whale versus shrews and mice, but one math formula can predict it. And here I apply this universal clock to humans and mice, dolphins, elephants, bats. So remarkably accurate. But let's drill a little bit deeper. So this colorful plot on the right shows you species sorted by maximum lifespan. So you see the dashed line in the middle, it's faint, but it encodes maximum lifespan. And so we start with short-lived shrews and mice, and then we go all the way to the bowhead whale. And the other lines that you see near the names of the animals, the solid lines, the jaggedy line, it's really the accuracy of epigenetic clocks, of the universal clocks in these different species. And I hope you notice there's no pattern, so the solid lines don't have a pattern at all. And that is simply the statement that a universal clock is as accurate in a short-lived species as in a long-lived species, And to me, there's something interesting going on. One could call it almost a biologic invariant with regard to maximum lifespan. And Vadim Gladyshev gave a wonderful presentation describing various interventions that affect really all epigenetic clocks, but certainly also universal pan-mammalian clocks. Caloric restriction in liver, growth hormone receptor knockouts, these dwarf mice or Snell mice, they show a slow epigenetic aging in all organs. Parabiosis is a nice finding from Bohan Zhang, Vadim Gladyshev. High-fat diet conversely accelerates aging, and various genetic conditions have been linked to epigenetic clocks. So why is it interesting? Because these interventions teach us something about the nature of the clocks. What do they measure? So yes, they relate to metabolism. Yes, they relate to hormones. They relate to intercellular signaling and so on. Now, what pan-mammalian clocks give us is a new era of testing. We can use literally the same biomarker, the same conserved biology to test interventions in multiple species at the same time. So let's say you have an exciting anti-aging intervention. You could really start a study in marmosets and mice, rats, and mini pig all at the same time using the same clock. Hopefully it helps you de-risk your preclinical studies. But there's one problem. Overall, epigenetic clocks are somewhat of black box predictors. We have, for example, 600 cytosines, and they predict age. And to really learn more about what are they measuring, we're using something called chromatin state analysis. That's a genome annotation pioneered by Jason Ernst at UCLA. And he uses histone marks to define different chromatin states. And to motivate that, let's start with some data. So here I show you a Manhattan plot where the x-axis shows you human DNA. But each dot is now a cytosine, ultra-conserved cytosine. And the x-axis shows age-related gain of methylation. The y-axis shows age-related loss of methylation. And you can just ask, well, where are these cytosines located that relate to age in all these species? And one finding is that they locate in regions that are repressed by a particular complex called polycomb repressive complex 2. This is a complex that represses certain regions in the DNA during development and cell differentiation. It's utterly important in development and defining the cell identity. But if you drill deeper, I show you here now different chromatin states. So age-related gain of methylation can be observed in PRC2 regions and what is known as bivalent promoter, bivalent regulatory regions. Again, these regions are very crucial for cell fate decisions and development. They show gain really in all cells and tissues. But when it comes to age-related loss of methylation, one needs to be a bit more careful. Yes, we find certain heterochromatin regions that show loss of methylation, but only in cells that proliferate, blood, skin, maybe liver, but not in brain or heart or skeletal muscle. So cells that don't proliferate don't have that pattern. Rather, other regions known as transcribed exon regions, they show age-related loss of methylation in tissues that don't proliferate, you know. All right, and here I just show you how these chromatin states that we are using a lot, the chromatin states are on the x-axis, and the columns correspond to many different tissues in mice, but also in humans, 2 species. And red means high methylation, blue means low methylation. And I think what I learned from this plot is that these chromatin states, the mean methylation levels are remarkably consistent across different tissue types, but also across these 2 different species, you know. All right. So moving on, I used to be a mathematician, and so I was always hoping that we could bring the power of math to aging biology. And I feel cytosine methylation gives us that opportunity. So here I show you some recent findings from a preprint in bioRxiv, fundamental equations linking methylation to maximum lifespan. And I'll start with an intuitive question. Does the rate of change in methylation relate to maximum lifespan in a species, right? The intuitive idea is if there is a fast rate of change, a fast increase in methylation, well, maybe the animal lives only a short life. And as a matter of fact, there are several articles that have been published in the past to address that question. And here we used the data from the mammalian methylation consortium to drill really deeply. And let me start out again with talking about these chromatin states that I just introduced. So that we have about 100 chromatin states, and every dot in this plot is a chromatin state. And then on the x-axis, you see how a rate of change in that chromatin state correlates with maximum lifespan in species. And what do I see? Well, I see that on the right, I see these bivalent promoter regions, these bivalent chromatin regions that have low methylation in most species. There, the rate of change relates inversely to maximum lifespan. But then also I see plenty of dots, meaning plenty of chromatin states that don't have a relationship to maximum lifespan at all, you know. So one needs to be very careful when it comes to this earlier equation, the rate of change in methylation versus lifespan. You need to really think carefully, where do you measure rate of change? And yeah, now if you pick bivalent promoter regions, then you do observe beautiful relationships with maximum lifespan. So here on the left-hand side, I show you how the average rate of change in methylation relates to maximum lifespan when you do a log transformation. And you see a very striking relationship. And each dot is a different species. So this is, again, the strength of the data set. We have here hundreds of mammalian species. We can rigorously test these questions. And on the right-hand side, it's basically not on the log scale. It's the original scale. So the y-axis, rate of change of methylation on the y-axis versus 1 divided by maximum lifespan. And you see a very nice relationship. So yes, but you know, let's think about it. When you calculate a rate of change, in a naive way, you really assume that there's a constant speed, that there's no acceleration or deceleration. You assume a constant velocity. But that's not the case. So here I show you that, as I mentioned, that methylation in general has a nonlinear relationship with age. So here we have blood samples from pigs from age 0 to age 6. And you see early on you have this log relationship and later this constant relationship. And you can define a rate of change during before puberty, so to speak, before the age of sexual maturity. That's the green line. And the blue line is the rate of change, the slope, in older animals. And what do you see? Well, early on, fast rate of change, later on, a lower rate of change in methylation. And here we do a formal analysis. So this is, each dot is now a species. And on the x-axis, I show you the rate of change in young animals before age of sexual maturity. And on the y-axis, rate of change in older animals. And I see two things here. First of all, as expected, in young animals, you have a faster rate of change than in old animals. But the second thing I see is there's a linear relationship. So interestingly, rate of change in young animals seems to have a linear relationship with that in old animals. Yeah, and so here I show you how, do you remember this equation that I showed you in pigs? But you can put all of our data together, really 10,000 different tissue samples from all these species, and plot methylation versus relative age. And then you get a messy plot, but you do see there's this log linear relationship. And so empirically, this is the best equation that we've come up with. But we are releasing the data because maybe it inspires some of you to come up with a nicer equation. And here I want to show you another finding. So here I sort species by maximum lifespan. On the top is the bowhead whale. This is the longest lived mammal we know of. At the bottom we have a shrew, shortest lived. And the bars correspond to the correlation between methylation and age correlations, in other words. And what do you see? The red line is kind of the average value. And there is no pattern again, So this comes back to my earlier statement that age correlations seem to be impervious to the maximum lifespan of the species. Mathematically, this statement of a very strong relationship between rate of change of methylation and lifespan is actually tightly linked to that statement of a correlation invariance. So in summary, I propose the following fundamental equations for methylation studies of aging in mammals. First of all, you need to focus on selected chromatin states and these bivalent regulatory regions. And then you do see this log linear relationship with age across the lifespan. Equation 1 is that the rate of change in methylation in these bivalent regions is inversely related to maximum lifespan. The second equation is that the correlation between methylation levels and age bears no relationship with maximum lifespan. And the third fundamental equation is that the rate of change in young animals is proportional to that in old animals. I want to now briefly talk about human biomarkers. Many of you have heard me talk about Grim age. It's been carefully validated by many people. For example, Cahal McCrory from Ireland wrote a wonderful paper. And we have developed a new version of Grim age. I want to alert you to it. It's slightly better than the original version, but it now also estimates C-reactive protein levels and plasminogen active, and hemoglobin A1C. So this Grim age marker is really a marker based on 1,030 cytosines. It doesn't use any plasma proteins, but interestingly, some of the changes can be interpreted in the sense that they do correlate with these plasma proteins. And somebody told me I should visualize how well Grim age predicts mortality risk. And here I show you different cohorts and different sexes. So, for example, Framingham Heart Study, males only, Women's Health Initiative, women only. So stratified by race and anyways, the blue curve corresponds to the 20% of people with the slowest aging according to Grimmage, and the red curve are the people with the highest, the 20% people with the highest Grimmage. And you see in most cohorts there's a pretty good separation in terms of survival time. So yes, Grimmage predicts human mortality risk. I've started a non-profit foundation with CEO Bobby Brook, and Bobby offers all of the methylation arrays that I talked about, in particular the mammalian methylation arrays, fully available to the research community. I hope you will use it. All of us are striving to bring down the costs. Roughly speaking, an array used to cost like $200 or so per sample. I wish we could cut that in half. So in conclusion, cytosine methylation allows you to build these pan-mammalian clocks. I had a joke, if you allow me. Let me try it out on you. So we built predictors of maximum mammalian lifespan, right? Now it's a supervised model. You have 348 species predicted. And so Cesar Lee in the lab developed this multivariate predictor. On a log scale, we can predict maximum lifespan quite well. This is a cross-validation estimate, correlation 0.89. So we have that max lifespan predictor, but I honestly have no use for it. I couldn't think of any application except for this one. So we looked at males versus females in different species. And we had, of course, hundreds of mammalian species. And what we found is that females have a mortality advantage in 17 mammalian species, except for one. And interestingly, of course, in humans, females have an advantage with regard to maximum lifespan. And so then what I want to say is, if you do want to increase your maximum lifespan, you have to become a woman. So there is no way around it. I can say that with a lot of confidence because we, of course, looked at all these interventions that we mentioned earlier. Does caloric restriction affect maximum lifespan? No. Why not? Because imagine you study 1 trillion mice and regular mice. Some of them will have the same max lifespan as caloric restricted mice, because it's max lifespan, not average lifespan. And similarly, we looked at many other interventions that you can think of, including parabiosis and so on. We don't see that these regular things that we know are healthy for us, they don't seem to affect maximum lifespan. Maximum lifespan seems to be set at birth. You get born with that, And then, so what's the new frontier? The new frontier perhaps is to move away from mammals. We published a small study where we and Vadim Gladyshev and others, there are 3 papers that describe epigenetic clocks for clawed frogs, the xenopus. And so we applied our mammalian array to xenopus, although it isn't designed for amphibians. And we even built a dual species clock that applies to humans and xenopus. But it's just very rudimentary. So I feel the future is extend the mammalian project to amphibians, to fish, other vertebrates, and so on. And I want to thank the Mammalian Methylation Consortium. I mentioned some names that you recognize: Vera, Andre, Vadim, Emma Thieling. And thank you so much. Yeah.
All right, thanks so much, Steve. That was wonderful, of course. We got time for one question before we, right there. You got the microphone still?
Yeah.
Yeah, I just had a question about the cytosine methylation. Do you see C to T transitions as a factor of age, driven by deamination?
I think it's a wonderful idea. You know, I haven't carefully looked at it, but I think it's very possible. Mm-hmm. Yeah.
Okay, that was quick enough that we have time for one more question.
Okay.
Didier, over there. Who's got the microphone? There it is.
Thank you. Extremely interesting. So a few years ago, you wrote an article about reversal of epigenetic age with treatment on thymus. It was 2 years less in 2 years, let's say. Also, I read sometimes other articles with a spectacular decrease of epigenetic age. But yeah, I'm convinced that, well, I'm not sure, of course, and I would love to be wrong, but my feeling is actually the people who had those treatments, they will not live 4 years longer, you know?
Yeah, that's true.
So it's like if epigenetic age is measuring age, but not when you change it temporarily, or can you...
Yeah, thanks for that question. Yeah, my main mission when I talk to journalists is to educate the public that they cannot translate a methylation age estimate into lifespan. It's a disastrous error, you know. So let's say you're a 50-year-old person, and then let's just hope your methylation age is 40, and then you jump perhaps to the conclusion, therefore I will live 10 years more than I should. That's really it's a mathematical error, a bad error, because so yes, we can predict lifespan, but it's a highly non-linear relationship. It's certainly not one-to-one, you know. I mean, to throw out some numbers, if you're 10 years younger in terms of methylation age, well, maybe then you live 3 years longer, you know. I mean, so just to reality check. And then also, for individuals, there's always a large error bar associated with a lifespan prediction, you know. That's why we don't report it, you know. So if I told somebody, you will make it till age 85, I would have to say to them plus minus 5 years, you see. So so I'm very uncomfortable with reporting lifespan. I want to briefly talk about the thymus regeneration trial. This was a study of 9 men, and now Bobby Brook and Greg Fahey have repeated this study with many more people and both sexes. And they have the methylation data. I got an email that the data have reached them. And so I guess next time I present something, I hope I give you an update. But I literally haven't looked at the data. Too much traveling, too much partying, you know, so. But yeah, thank you.
Yeah. Well, so one could say that we never have too much partying, but first of all, applause for Steve.
Thank you. Hi.